In the words of Vincent Racaniello at Columbia Medical School, we live in a virtual cloud of viruses. We eat them, we breathe them. They are everywhere, even in our genomes. For example, in July 2017, scientists in the laboratory of Terry Heidemann at Tufts here in Massachusetts reported the presence of an unusual protein in the blood of pregnant mothers. This protein, called HEMO or HEMO, is made by human fetuses from a gene that originally came from a virus that infected our mammalian ancestors over 100 million years ago. In fact, there are approximately 100,000 pieces of rogue viral DNA in our genome. Remember how we said that the human genome is approximately 98% repetitive and/or junk DNA? Well, 8% of that is viral. Yes, viruses are everywhere, in the air, in the soil, in our food, in our water, even in our oceans. But what is a virus? In this series of videos, we'll consider that question and more. Let's get started. Our goals for this video are pretty simple and pretty basic. We just need to be able to describe what a virus is in terms of its structure and characteristics. So let's start with the definition. Viruses are small, non-living, obligate parasites that replicate only inside of cells. Infected cells, and by the way, infected organisms, are called hosts. Scientists think that viruses evolved along with living organisms, or possibly they existed before living organisms did. Why? Well, primarily because viruses infect all forms of life. You heard me right. There's a virus for pretty much every living organism on the planet. That means that protozoa can be hosts, animals can be hosts, plants can be hosts, fungi, yep, they can be hosts, and bacteria have a number of viruses that infect them also. Believe it or not, there are even viruses that infect other viruses. I know, check them out. They're called virophages. Now, let's look at the parts of this definition in a little bit more detail and briefly talk about some of the characteristics that distinguish viruses from living organisms like bacteria. You may be wondering, what is an obligate parasite? Well, let me explain it with an experiment. If you were to take a petri dish of nutrient agar and plate millions of bacteriophages, that is, bacterial viruses, on its surface, then incubate the plate and measure how many bacteriophages were on the plate after 24 hours, you would find that there were the same number or maybe fewer bacteriophages than you had put on the plate the day before. Now, if you repeated that experiment, but before adding the bacteriophages to the agar, you mixed them with the right bacterium, say E. coli, and then place the E. coli and the phages on the petri dish, incubate for 24 hours, you would see something like this, the image that's on the right side of the screen. Now, the turbid background, kind of like what you see here, it looks kind of whitish and cloudy. That's where bacteria, E. coli, are happily growing and undergoing binary fission. As they grow, they make a film. You've seen this before when you've looked at colonies in the lab, but they make a film on the surface of the agar. The clear circles, like the ones right here, represent events where when you mixed the bacteriophage with the bacteria, a phage actually attached to a bacterium and began to inject its DNA into that bacterial cell. As the bacteria were plated onto the agar, the genes from the bacteriophage start to take over the bacterium and turn it into a virus-making factory. Eventually, after some hours, the viruses will begin to assemble, and these cells, these bacteria that are full of new viruses, will be lysed. virus will spill out into the environment where it will go on to infect adjacent bacteria to that site, and so on and so on and so on. So you're left with an area that looks clear on the plate where the bacteria have basically burst open to release viruses. This phenomenon does not happen in the absence of cells. Viruses cannot grow without a host, so they're obligate. Now, in order to make more copies of themselves, viruses essentially have to take something from their hosts. They have to take nucleotides, amino acids, energy, etc. And they harm their host as a result because the host can't continue to grow. And in the case of phages like what we see on this picture here on this slide, sometimes the host actually dies. So they are obligate parasites because they have to have a host and they hurt that host. So during a virus infection, a virus like this influenza virus has to gain entry to a host cell. This usually happens when a virus attaches to the host via a protein or carbohydrate on the surface of the host cell. Then the virus is brought into the cell in a vesicle. Next, the virus falls apart, releasing its nucleic acids. Then the magic happens. The virus will usually turn off or reduce its host's ability to make cellular DNAs, cellular RNAs, and cellular proteins. Instead, the virus and its proteins will reprogram the cell to use cellular polymerases, ribosomes, and other molecules to make viral nucleic acids and proteins. That's essentially the parts needed to make new viruses. These parts will then assemble somewhere in the cell, at a membrane, for example, or in the nucleus or in the cytoplasm. And then the virus will either bud away, like in the case of influenza, or the cell lyses. This is different from binary fission, isn't it? Remember, bacteria mostly, but not universally, reproduce by binary fission. In binary fission, a cell reproduces its own DNA and then splits itself in half to make two new cells, or in this case, two new bacteria. With viruses, by the way, one reproductive cycle can make anywhere from tens of new particles at one time to over 50,000 at one time. In the case of some viruses, they will make millions of new virus particles over the lifetime of their host because they don't immediately kill their host. They live within it for some time. Sometimes the virus can live within its host for decades. Think about HIV, for example. Now, the definition for viruses also said that viruses are small. If you thought that bacteria were small, viruses are super tiny. The image on the left shows an E. coli cell. You'll recognize the bacillus-shaped cell here. So this is an E. coli cell that is being infected by a bacteriophage called T4. So all of these viruses like this, they look kind of geometrical on the top, and the geometrical part is standing on like a head or a pin, like this. Each one of those structures is a virus that's trying to infect this bacterium. Now, by now, in lab, you've looked at bacteria under the microscope. You may have even seen E. coli, so you kind of know that these cells are really small. This strain of E. coli has dimensions of about 5,000 nanometers by 500 nanometers, so that's length by width. The viruses that you see in this image have dimensions of 200 nanometers by 90 nanometers in width. And this is actually a large virus, so I'm showing you this picture so you can see how small viruses are in comparison to a bacterial cell. And again, remember, this is a large virus. Most viruses, or many viruses, are only a tenth of the size of the ones shown here. A good number to keep in mind is that on average, viruses range anywhere from 20 to 200 nanometers in length and width. The picture on the right here shows HIV. So the little green specks that you see here, those are HIV viruses that are trying to infect a cultured T lymphocyte. Lymphocytes are eukaryotic cells, and eukaryotic cells are something like 50 to 100 times larger than E. coli are. So as you can see in this picture, the HIV particles are really small compared to the blue lymphocyte. HIV has an average size for a virus, it's about 120 nanometers, so roughly half the length of the bacteriophages in the picture on the left. Until about 2003, we thought that all viruses were super tiny. Then a curious microbiologist happened upon the first of what are called giant viruses. These viruses are on the size of some small bacteria and archaea. Interestingly, they can contain genes for amino acid and nucleic acid synthesis, suggesting that they may be a fourth domain of life. One would think that these viruses were pretty rare, since we've only known about them for 15 years. But in actuality, it's possible that we've actually misidentified things that we thought were bacteria when they are actually viruses. So just to give you an idea of how big these giant viruses can be, this figure shows you an E. coli cell taken with an transmission electron microscope. These viruses, the pithovirus and clothovirus, are kind of the same size, so it's easy to understand how scientists might have misidentified certain viruses as bacteria. They even have bacteria-like shapes. The last part of the definition states that viruses are non-living. If you think back to your college bio class, you'll remember talking about the characteristics of living things. You learned that all living things are composed of one or more cells, and that cells are the smallest unit of life. Cells are membrane-enclosed entities that maintain a stable internal environment via a process called homeostasis. They have a DNA core, like you see here, that is surrounded by cytoplasm and a plasma membrane, which is the yellow layer, you'll remember. Viruses, in comparison, are not cells. Although viruses are somewhat self-contained, and some even have lipid layers that resemble plasma membranes, these are not cells. They do not perform homeostasis, they do not have a cytoplasm, they do not contain organelles, not even ribosomes. Structurally, viruses consist of a nucleocapsid and sometimes an envelope. I'll describe the nucleocapsid first. All viruses have a protein shell called a capsid. That's the blue geometric structure that you see here. This shell is made up of small subunits called capsomeres, represented by the blue spheres. This shell's job is to protect the virus's nucleic acid, either DNA or RNA, but never both, at least not as far as we know. The purple squiggles here represent the nucleic acid, so you can see that's packed inside of the capsid. Now, this nucleic acid may have proteins bound to it. There can be scaffolding proteins that help to orient the nucleic acid inside of the capsid. There may be virus-specific polymerases that will copy the nucleic acid during an infectious cycle. There may also be other replication support proteins that help turn off cellular proteins and allow the virus to replicate inside the host. Altogether, the nucleic acid, the capsid, the scaffolding proteins, and any replication support proteins, they make up the nucleocapsid. Nucleocapsid is spelled Nucleocapsid. Nucleocapsid. Now, as some viruses leave an infected cell, they snatch a bit of the cell's plasma membrane, and that becomes part of the virus's structure. This bit of membrane is called an envelope, and it usually has viral glycoproteins called spike proteins, which are necessary for infection, by the way, embedded within the envelope. This figure shows you the layout of an enveloped virus. Rising up from the viral surface, you see these red spike proteins. They are embedded in a lipid bilayer, which is represented by the green here. This is the envelope, these two things together. Deep to the green layer, so if we slice the virus in half here, we can see that there is a teal layer just under the envelope in green. This is the capsid. Deep to the capsid is the nucleic acid for this virus. In this case, it's RNA. Another important characteristic of living organisms is that they are highly organized and complex. This is most clearly observed when small molecules organize in specific ways to make larger ones. We definitely see this with viruses. They have nucleotides that come together to make nucleic acids. They have the programming to help amino acids assemble to make proteins, and we even see it in virus structures, for example. Viruses are primarily classified into two groups based upon structure. The helical viruses usually have elongated shapes. They consist of one or more strands of nucleic acid, which is represented by the red strands here, surrounded by capsimers that form a helical arrangement around the virus, making the capsid. Examples of this type of virus includes the rhabdovirus, which you see here. This is the bullet-shaped virus that causes rabies. Tobacco mosaic virus, that's this one. It looks like a matchstick. This was the first virus ever discovered. Filoviruses, which are called the twisted threads because of their appearance, like Ebola. And then the pleomorphic influenza viruses, like this one, that look like gumdrops or sometimes spheres. They can have multiple shapes. The other major structural class of viruses is called the icosahedral or quasi-spherical virus. It's based on a shape called the icosahedron. An icosahedron is basically a solid object, like this one, that's built of 20 identical faces, each of which is an equilateral triangle. Examples of icosahedral or quasi-spherical viruses include human papillomavirus, which looks like this, and poliovirus, which looks like this. You also have some complex capsid forms that include this icosahedral arrangement, like that of your bacteriophages. Another basic characteristic of all living things is that they reproduce new organisms like themselves to carry their genes, and they'll hopefully pass those genes on to a new generation. Viruses definitely do this, and they can do it quickly. It depends upon the virus. Rhinovirus, which is the cause of the common cold, takes up to 3 days to make enough virus particles in the human body to cause symptoms like fatigue and sneezing. Unlike living organisms, however, viruses don't grow bigger per se, and they don't develop. Once a virus is assembled in its final form, it will not change, unlike a baby that constantly changes after birth as it grows and develops. While we are considering viral replication, we should consider evolution. Viruses, like living organisms, do evolve over time. typically at a faster rate than for living organisms. This, by the way, is why you need to get a new flu shot every year. In living organisms, there are many mechanisms of genetic variation, but I've grouped them into three classes. You have, number one, mutations. These are changes that arise when mistakes are made during DNA replication. You have, number two, horizontal gene transfer. And number three, phenomena that are related to sexual reproduction. These mechanisms provide variation in offspring that may make them more suited or better suited for survival in the environments they live in. Thus, they are more likely to pass their genes on to future generations. If the changes are not advantageous, on the other hand, then those offspring may be more likely to die prematurely or be killed, possibly before they get to pass their genes on. Over many years, these changes will accumulate in a population of living things and change their phenotype slowly. Viruses evolve more rapidly than cellular life does, sometimes within a few months to a year. Viruses evolve two ways. Number one, through small mistakes that are made as their nucleic acids are replicated or copied. These small mutations can lead to changes in a virus structure. So for example, in this diagram, we have a spike protein that looks like a purple circle here, but that mutates to become a half circle, like you see here. These types of mutations may make it easier, for example, for a virus to infect a host, or maybe to leave that host, or maybe those changes can make the virus more resistant to antiviral drugs. In any of these cases, this type of mutation would be beneficial. I should also note, however, that some mutations, because of the spontaneous nature of how they occur, some mutations will actually be detrimental to the virus. A second way that viruses can evolve over time is through recombination and reassortment. Recombination and reassortment can occur when a host is co-infected with multiple types of viruses at the same time. This recombination reassortment is responsible for the appearance of a new influenza virus in 2013 called H7N9. According to the CDC, 8 genes for this new virus are related to avian influenza viruses found in domestic ducks, wild birds, and domestic poultry. The idea is that when a single host was infected by 2 or more of these viruses, like you see here, recombination happened so that genes could be swapped among segments of RNA, producing new gene combinations. In addition, the actual RNAs may have also been mixed up. So as you can see here, we have 3 viruses. We have H7N3 in domestic ducks, we have H7N9 in wild birds, and we have H9N2 in domestic poultry. When those recombined, we get a gene mixture that looks like this. So the blue gene here on top came from the domestic duck virus. The green gene right here in the middle came from the wild bird virus. And then the domestic poultry contributed the remaining 6 genes. This combination of genes produced a new virus to which humans had no immunity. So the reason why this particular virus was of concern is because, number one, it's a new virus. There's little or no immunity to it. And number two, most infections with this virus resulted in severe respiratory illness. The mortality rate was approximately 30%. Fortunately, this virus doesn't appear to be transmitted from human to human yet, but should it evolve to do that, we could be looking at another pandemic in a few years. Finally, two major characteristics of living organisms is that they respond to changes in their environment, number one. Remember back in college bio, we called these changes stimuli. Living organisms respond to the presence of nutrients, toxins, light, temperature changes, pH changes, pressure, all sorts of different things depending upon the organism. Viruses don't respond in the same way. They're more likely to be affected by changes in their host than conditions per se. Let me give you an example of what I mean. If we take an E. coli cell that has a bacteriophage in it, remember that's a bacterial virus, and we shine UV light onto that virus, onto the bacterium, the virus will actually cut itself out of the bacterial chromosome and it will lyse the bacterial cell because of the UV light, but it's not responding directly to the UV light. What it's responding to is that as UV light shines on the E. coli cell, it causes thymine dimers, which damages the cell's DNA. As the cell tries to repair the DNA, the same proteins that are responsible for repair will actually trigger the bacteriophage to cut itself out of the chromosome. So it's not directly responding to the UV light, but rather it's affected by what's going on in the host. So it's not exactly the same thing. The second characteristic of living organisms is that they make and use ATP to perform cellular work. And viruses, frankly, don't do that at all. They have no way of taking in nutrients and turning that into ATP, and they have no way of taking that ATP and doing cellular work. For the last few slides, I've gone over some of the important characteristics of living organisms in order to contrast them with viruses so we can see how living organisms are similar to viruses and how they are different. On this slide, I want to summarize what we said. We said that viruses are non-living particles. The fact that they're non-living distinguishes them completely from living organisms. I am keeping tally of similarities versus differences using red X's. The red X here means that these two things are not the same. Living organisms and viruses are not the same with respect to being living and non-living. We also said viruses are acellular. Living organisms are composed of at least one cell, so that's another big difference between these two microbes. Viruses are obligate parasites. It turns out that there are some prokaryotic organisms that are also obligate parasites. They must live inside of another host organism. So I put in equals with a slash for this because it's not exactly the same. The viral life cycle is not the same as the life cycle that we'll see with those parasitic prokaryotes. So that's represented by the equals with a slash. We said that viruses are super tiny, and they are. Your average size was between between 20 nanometers and about 2,000 nanometers in size, with 2,000 nanometers being around the size of your smallest living organisms, your smallest bacteria. Then I told you that you have the giant viruses that have been discovered within the last, 15 or so years. So, viruses generally are super tiny, but then there's the exception of the giant viruses. So that's why there's an equals with a slash there. Viruses are extremely ordered structures. I showed you different images of viruses, and you learned that they have capsomeres that come together to make capsids that surround nucleic acids and may or may not be enclosed in an envelope. You also learned that viruses are classified into two groups based upon their structure. You have the helical viruses and you have the quasi-spherical viruses. So the order that we see in viruses is similar to what we see in living organisms. The amount of order that we see is similar. So that's why that gets a green check. Viruses reproduce themselves. They make progeny, that is offspring, that contain their full complement of genes. And then those viruses go on to pass on their nucleic acids as well. So from this standpoint, viruses are the same as living organisms. The goal is to reproduce. So they get a green check for that. Viruses are unable to grow or develop. Remember, once they're assembled inside of a cell, that's it. They're not changing, they're not getting bigger. So that gets a red X because living organisms may grow and develop. Viruses do evolve over time. It happens at a faster rate and through a few different mechanisms, but it's still evolution. And in that way, they are similar to living organisms. Viruses are unable to make or use ATP. They have no metabolism, no metabolic activity just by themselves. So that gets a big red X. It's not the same as living things. And viruses do not directly respond to stimuli. So that's another big red X. So if you look at the symbols here on the left, would you say that viruses are very much like living organisms, or are they very different? That's it for this video. I hope that this is useful to you. This material, all of it is testable. You could see this on exam number 3. So you want to be very familiar with this material. I'll see you online.